easy and you should be able to understand it. Don't feel that you should be able to understand it all. A lot of it, I'm just going to be introducing some, basic biology and chemistry, because you kind of have to go through that before you understand what we understand sort of outside the Earth. So there's a field out there today that's called astrobiology. It's a very funny name for something. As I told Bill, it's an aspirational name, right? Chemistry, we study substances. Biology, we study life. Astrobiology studies life outside of Earth. And of course, there is no known life outside of Earth right now. So it's kind of odd, right, that your discipline is named after something that is you don't know exists yet. But it's out there. So actually, when I was in grad school, I did get to go to NASA's big astrobiology conference, not too far from where I live now, which was kind of funny. And it was just a cool, just amazing place. You got all these people from many different disciplines: geology, astronomy, biology, chemistry, all together under one tent. It was held at Moffett Field, where they had these big hangars that they're just gigantic, and just a group of very smart people. You won't get that many different types of people under one tent very easily. So it was just a cool thing. And when I went there, I learned a lot about it. And so I'll just sort of go through what we know. I'm using the word fiasco here. That's a part of astrobiology, and you're going to see why it's like that. It's such a difficult problem to figure out what life is out there, that it seems to invite sort of the most terrestrial behaviors we know amongst humans. And so we're just going to take a look at that, and let's we'll get started. So just a quick primer on biology and chemistry. We'll go through. What I'm trying to do through this presentation is actually give you hopefully some tasty facts, that things that are fairly recent knowledge that I hope you can sort of remember as like, oh wow, that's relatively new. One thing that you may not know, that you may, whatever you remember from biology textbooks, not too long ago, is that the tree of life. What is the evolutionary tree of life? So until recently, the accepted sort of tree of life was that there were three domains: bacteria, archaea, and archaea is a specific type of microorganism. If you just took a picture of them under sort of standard imaging techniques, you and I couldn't tell the difference between them and bacteria. They look just like bacteria, but they're totally different. Their lipids are different. Their internal machinery is quite different. So they're a very distinct branch of life. They used to be called archaebacteria. Now they're just called archaea. And then eukarya, which is the root of all complex life: protists, plants, fungus, and animals. One thing that's an interesting thing to note is that, we think of fungus like mushrooms and other things like that. You always think of them as, at least I do, I always think of them as sort of like plants. Fungi is actually is evolutionarily closer to the animal kingdom than it is plants. That is, fungus and fungi and animals. I'm sort of mixing up the names. I should have said animalia and fungi, I guess, but they have a common root, which has a common root with plants. It's not that plants and fungus have the same root, kind of thing. So that's kind of interesting, that fungus is evolutionary closer to animals than plants are, or closer to animals than plants. What has come about through very relatively recent discoveries is evidence from archaea that they found sort of, I think, somewhere off the coast of Scandinavia, and doing some deep sea diving, and they've given these archaea names like these like Scandinavian mythology names, like Odin and like all these gods from Scandinavia. We think now that life actually has the tree of life has a two-domain structure. That is, that bacteria, I should tell you here, sorry, I didn't mention, Luca is the last universal common ancestor. So the root of the tree is the origin of life. We don't know how that started. That presumably evolved in some way, some sort of protocell that was doing something until a last universal common ancestor. And that's the root of the three domains. What we think now is true is that there are two domains to life. That is, from the last universal common ancestor, one branch went to bacteria and the other branch went to archaea. From archaea, all modern complex life evolved. I shouldn't say modern. We're all the bacteria and archaea are still here, but all complex life evolved. That's profound. Two domain structure, and that's something that's relatively recent. It was always been suspected, but the evidence for that has become stronger only in the last 10 years or so, which is quite interesting. The other thing you may know about archaea is that archaea, you can get bacterial infections, you can get fungal infections. There's no known archaeal pathogen. That is, archaea don't interact with us in the same way as far as we know. They're around. You've got them in your gut. They're just around, hanging around. They're tougher to work with in culture. Oh, did you say something, Bill? Sorry, did I miss that? Oh, okay. So they're around. So anyways, that's the tree of life. So when we're talking about astrobiology, we're trying to figure out, there are many more branches here, of course, that I haven't shown. There are many different types of animals, fungus, plants, etc. But what we're trying to figure out is, what this looks like in other places. The key thing to know about life is that life runs on carbon chemistry. Everything we know about life is centered around the chemistry of carbon molecules. That is, molecules that are mostly made of carbon. And to sort of emphasize that, if we go back to, if I get a chat, just let me, I might miss it if it, oh, there we go. Okay, Thailand, got it. Yeah, if you just want to speak up and interrupt me that way, I can just address there, because I might not be able to get to the chat.
Okay, Maneesh.
Yep.
I think what we should do is let you go through your presentation, and anybody that wants to
oh, sure.
kick in afterwards can ask questions. Okay.
Sounds good. Let's do
if that's okay with you.
Yeah, absolutely.
Okay.
So this is a really important, in 18, I can't even see the thing here. There we go. In 1828, Wohler, I'm probably not saying the German correctly, came to a profound insight, and that was that things that we associate with life don't need to come from life. That there's nothing, there's this theory of vitalism that his teacher had sort of was a proponent of, and that was back in 1828. We didn't know. We didn't really have a terribly coherent understanding. I mean, people are a lot smarter back then than I think we give them credit for, but you didn't have all the advantages of modern instrumentation and modern scientific understanding. And so what he did was he demonstrated from salts of the earth, this is lead cyanate, this is ammonia, this is water. If you mix these things together, you can get the molecule urea. Urea is the main component of urine. The urea doesn't smell like anything. It's the breakdown of it and other things that are in urine that make things smell when you walk through an alley late at night. But what he showed with this synthesis, and I sort of drew it out completely so that you could see it. This is lead cyanate, and if you put it with some, and I balanced everything out, so it's like these are the two ammonia molecules, these are two water molecules. The next step is here. You have an ammonia and a cyanate ion. The lead is over here. Don't worry about the lead; it's not important. And these two things, this hydrogen, the proton pops off onto here, and then this goes to urea. And they knew that substance that they got at the end was urea, or Wohler did. And that was the end of vitalism. He showed that you could get a substance that we had associated with biological organisms that you could only find in the urine of animals.
Okay, Maneesh.
Yep.
I know I said leave all the comments to the end,
sure.
But you're talking urea. My question is that like anything to do with the urea formaldehyde or whatever we have in the installation here? Is that the same sort of thing, or?
Yeah, I mean, urea is a chemical that's around and used for things, and it's very similar to formaldehyde. If the thing that would make that formaldehyde would be two H's instead of the N's there, and so it's a substance that's a precursor to many things, and it's a standard organic chemical.
Okay.
Yep. And
so I apologize for breaking in there, but
no, that's okay. That's good. Yeah, I think someone. I think if something's not clear or just please just I think a quick break-in is. I know it's a little late, and we'll try to get through this fast, but I think a quick break-ins are totally reasonable. So that was just a profound insight, right? This showed that there's nothing. And if you were alive at that time and you're a critical thinker, you could just imagine that. That's right. I bet you could make everything that's in an organism from there's nothing magic, right? There's no vital force present in the material that makes up living things. It's just stuff like everything else, like the rocks around us and everything like that. And that was definitely the thing that ultimately this led to organic synthesis. This led to the dye industry, and the dye industry is what led to the drug industry. And that's where we are today. And you're going to see this type of knowledge, how far we've taken. It's really, really impressive. And you may ask, well, why carbon chemistry? What's so special about carbon? Well, it's not so if you take a look at the periodic element table, most of this, the parts that sort of we say that are most obviously recognizable as life are these purple ones here. These are the non-metals. A lot of other things in the periodic element table are very metallic. These are alkaline metals. These are transition metals. These are metalloids. And so it's just, these are the metalloids here. These are other metals. So most elements are pretty metallic, right? These are, of course, gases and temperatures over here, but it's only this little purple triangle that these are sort of the elements that we sort of most strongly associate with life. Carbon, nitrogen, oxygen, phosphorus, sulfur. And even selenium is used in there. I just sort of, it's not that we don't use other elements, we do. I just circled a few more here. You're familiar with a lot of them. Calcium, that's what makes up your bones, right? Calcium carbonate, iron, you can't get very far in life without any iron. And all these other ones, most others are trace amounts, even selenium, and I'll show you where that's used in us at least. Iodine, you wouldn't think that iodine is, of course, everyone knows iodized salt. That's what you get at the grocery store. And because if you don't have any iodine in your diet, you will get gout. So you need that for a specific hormone. So we need lots of other things. It's not that we don't use these other metals and a few other elements, but the main ones that make up the structural parts of us, that all carbon based, and mostly this corner here. People argue about other elements being important, trace metals. It's almost impossible to tell. For instance, silicon is everywhere, sand, right? It's the ground. We don't know if that's an essential element or not. We just can't answer the question, can we survive without silicon? Because silicon is everywhere. It's certainly not used in any obvious way inside us, but whether you could survive without it is almost an impossible hypothesis test, because there's just so much silicon around. So this is an example of what these small molecules look like. These are the carbon molecules that you're probably all familiar with. And what you can see, that's always where I don't draw, where there's a little vertex, that there's a carbon atom there. The standard way of drawing in chemistry is you don't put it there because there's so many carbons, as you can see. So when we think of organic molecules, they are essentially, chains of carbon with other little atoms sort of peppered around. I've just shown you nitrogen and oxygen here, but this is a standard thing in organic chemistry, or rather common organic molecules. I just labeled something here. This is ethanol. You drink that in a drink. Glucose, estrogen is a hormone that you're familiar with. Vitamin A. This is the molecule that, if you remember from the last presentation, this is what's sitting in the middle of that opsin protein that is helping you detect color. Indigo, this used to be a molecule that was farmed in colonial operations in India through the plant and extracted. And then once we figured out organic synthesis, what started with what you saw with urea, that was all shut down. You could make this stuff in vats, and so you didn't need to grow a giant colony of plants and extract indigo from them. Dopamine, you've seen. And these ones on the bottom, again, you're probably familiar with. Caffeine. This theobromine, that's the active ingredient in chocolate that gives it psychoactive properties and its taste. You can see how similar they are. Caffeine and theobromine look pretty similar. You got this pentagon here and hexagon here with some nitrogen atoms and a few things hanging off of it, and they are indeed similar molecules. The last one is nicotine, right? What's in tobacco smoke, or what's in tobacco leaves, rather. So these are, you've heard of all these molecules and stuff like that. This is what's sort of floating around in our bodies, and this is what they sort of look like. They're carbon skeletons, and carbon is the only. It's not that life has chosen carbon out of a bunch of other elements. Carbon is as far as we know, under these conditions that we're familiar with, carbon is the only element that makes these kinds of little structures with hexagons and chains and lines in this sort of discrete way. No other element works quite this way, as far as we know. We can always put a few other elements in there. We can pepper a carbon skeleton with nitrogens, oxygens, and things like that. But the sort of underlying structure, you can just see it's mostly carbon kind of thing, or at least the main chain of it's carbon. And that's a unique property of carbon, and that's what we think life is. Life as we know it is based on this carbon chemistry. The other important molecules in life that you've heard of, DNA, right? you've all heard of A, C, G, and T. This is what these nucleotides look like on their own. There's a sugar base, that's ribose, and DNA means deoxy. So whereas there's an hydroxyl group here with this oxygen, in RNA, there's another oxygen group there, but DNA is deoxy, so it's one oxygen short. And they all have the same ribose sugar, and then they're attached to a nitrogenous base. And this is adenine, guanidine, thymine, and cytosine. And so they all have different structures, and the DNA is double-stranded, and you may have seen this before. There's what we call base pairing. A pairs with T and G pairs with C. So I've sort of drawn that out here. And these dotted lines represent what we call hydrogen bonds. There's a little bit of a negative charge on the oxygen. There's a little bit of a positive charge on this hydrogen. It's overall neutral, but each of these atoms sort of pulls on electrons with different strengths. So these are a little negative, that's a little positive, and you get these bonds. And so this is characteristic, this is base pairing. This is what, as far as we knew, is what enables DNA to recognize itself, right? So you have two strands and they fit together. A will go with T and G will go with C. And they won't form very strong bonds with any other thing. When your cells divide and when they replicate, that's how DNA is copying itself. That's how it can do it, by matching those up. And that's how it avoids making errors. That's why when your cells divide, they get what we understand to be very accurate copies of the DNA between the two cells. And that's how that works. And and I should also point out, your DNA is a chain of these things, and the chain is connected by this phosphate group here. So you can see where it's free here in the sort of free nucleotide, in the residue, they just sort of connect together through this phosphate backbone. And that's important for something we'll talk about soon. These DNA bases encode what they do, and I'll show you a diagram that's going to make it clear in a second. DNA is sort of the software of the cell, and what that DNA is doing primarily is coding for proteins, which are chains of these types of molecules, which are amino acids. And there's 20 of them. They each have a letter. And the only thing that we'll just mention here is that you can see what's common. They might be looking a little confusing right now, but there's a common chain here. To see this, there's the carboxylate group here with two oxygens and a carbon, and then the alpha carbon, and then that's the amine. So they all have that same pattern. That's what makes an amino acid. And then there's just something else sticking off the end of it. And that side chain, as we call it, has different chemical character across the different amino acids. Some side chains are very oily and they don't like water. Other chains are very polar and they're happy to be exposed to water. And what happens is when you make a long string of these things and you hook them together in the right way, that you pop one hydrogen off and you pop that OH off, and then you draw a bond together between them, they form a protein chain. And that protein chain folds in water to form a structure and do all the miracles in life that we know. If you remember the last talk when we were talking about the opsin proteins that are responsible for sensing light, that's a protein chain with a vitamin A molecule stuck in the middle. And that structure has to fold that way to do what it does and ultimately give us the power of vision. And every single, we're all through the pandemic, you guys have heard about spike protein now, I'm sure. You've heard about all the, things that weren't as prominent in the public discourse before. Spike protein is a protein just like that. It's a long chain of these things. And so this is sort of the hardware of life. And this is just sort of what a protein chain might look like. You can see I've sort of hooked the ends, the nitrogens to the carboxylate groups, leaving one off, and the side chain sort of hang off the side. So this is just in 2 dimensions. And what happens, with a real molecule, it's a floppy chain. And based on the character of these side chains, some of them will bond with others. Some of them will, the oily side chains will want to sort of talk to themselves and expose the water-loving side chains to the outside. They fold into structures, and those structures are what sort of make proteins function. All these amazing things, like your antibodies are proteins, your light receptors are proteins. Everything that that's doing something amazing is a protein in your cells. And if you can't draw out the individual atoms because these chains, they can be hundreds, thousands, tens of thousands, units long, lengths long. And this is just a picture from the protein data bank where all these structures are stored. And an artist, Arthur Goodsell, makes these sort of pictures that are really well known. And so if you imagine that the skeleton I sort of showed you, imagine it's sort of paper mache. There's just so many atoms moving around. You have to think of it as a paper mache over it, because atoms aren't little dots. They have a surface. So if you draw a sphere around all these things, it's really sort of like a bubbly sort of thing with a smooth surface. And you can just see this zoo of shapes in life. And these are the shapes, these are some of the proteins we know. And I can just briefly recognize some of these. This is a DNA strand. This is a polymerase reading off this DNA strand. It's probably making RNA from this DNA. There are transcription factors on your DNA that control which genes are on and which genes are off. They're like sort of traffic cops sitting there. These little balls here, these are certainly viruses, virus capsids. They're really well known. They have there's little balls of protein that do what they do. This too. There would be hemoglobin, as you know, carries, has an iron atom inside it, and that's what carries oxygen to all your cells. That's how you're alive. I think that's a tRNA molecule, looks like to me. So these are the parts that your cells are made of. This is what they actually look like. It's that form that allows them to do what they do. And so that's just a really quick look at a bunch of them. That you have tens of thousands of genes inside your cell, and you have tens of thousands of different types of these things, but all these things are sort of bumping around and moving against each other to make your cell operate. That's what they look like. It's a pretty profound picture. And this is just a standard sort of textbook look at how you get from nucleotides, DNA, to RNA, to proteins. You have the four different types of bases. They come in a sort of sequence. They're in a helix. There's a protein machine, a protein RNA machine that'll read them 3 at a time. And each 3 letters correspond to a specific amino acid. So the first one is GCU. So you can, this genetic code, so GCU, that's alanine. So that would be this amino acid here. And then the next letter is the same way. This gets read off and made into a long chain. What's profound is that this genetic code is essentially universal, whether it's a bacteria or a rhinoceros. Same genetic code that's running. That's pretty crazy to think about, right? These very, very different organisms use the same genetic code, and that's how we know we're part of the same tree of life. So that's another sort of big thought to keep in mind. The one thing I didn't talk about too much about all these molecules is there's this notion of chirality. We can talk a little bit more about it, but it's basically you can have 2 molecules that are essentially like your left hand and your right hand. Your left hand and right hand are kind of the same. I'm just holding them up right now if you see my picture down there, but they're not superimposable. No matter what I do, I can't match my left hand with my right. They're the same sort of shape, but they're the only way I can match my left hand and my right hand is by putting them through a mirror image. And because they're chiral, can't quite do it. So molecules are like that too. Every time you have a carbon with 4 different things hanging off it, that carbon is a chiral center. And that chirality, there's 2 different, depending on what your chirality is, you'll have different 3-dimensional shapes. So this is aspartame. You get it, in your sweetener packs that are out there. It has a sweet taste. That's what we call the L form with this chirality. If I make the molecule with the opposite chirality, it's actually bitter. Won't taste like it because we're all our proteins and all those DNA bases. You can tell when something's chiral when you see a lot of these, you see any of these wedged and dashed lines. That sort of indicates chirality. So all our amino acids are chiral, all our DNA bases are chiral, and this is one of the big puzzles of life is homochirality. That is, everything has the same chirality. Now it's easy to believe that we have the same chirality as our whatever bacteria or archaea that we descended from, because if you're using that chirality throughout, you can say, well, then all your descendants would use the same. But anytime you make molecules in a flask, you're always going to get 50-50 without any chirality there, because it's just they're the same thing. So you can't get more of one hand than the other typically when you're mixing things in a flask, because there's nothing to sort of break that symmetry. You're always going to get 50-50. So how life ended up being homochiral is a big puzzle. It's a very, very prominent feature of life. Was it just a frozen accident that however life started, that you were sticking, that molecule just got lucky and then that just kept going and won? Or is there some other reason? Is there some other asymmetry that that rests on? We don't know. So now you know just as much about chemistry and biology as I do. So now we're going to talk about some what I call fiascos, and it's just to sort of whet your appetite. And this is just quick fun. Does anyone recognize this picture?
Yes.
Off the top of their head? Anyone want to say? I had to look it up, but when I saw it, I remembered it very well.
That was a sign of life that got out of a rock on the Antarctica.
You got it, Mr. Clayton. You got it. I'm sending a chocolate over to your house. So
Oh, thank you.
Good job, Magic Merlin.
So yeah, this is Allen Hills meteorite, found in Allen Hills, which is a place in Antarctica. Landed on Earth 13,000 years ago. I remember this announcement. This was in 1996.
Yeah.
And I remember this picture, and I remember looking at it, and I remember the news too saying these were microscopic fossils of bacteria. And if you look at it, and scientist tells you that, you're like, sure, I've never seen a fossil of bacteria. I presume that's what they look like, and you guys know what you're doing. David McKay, I think was his name. I hope I know his first name wrong. I think I've written on the next slide. And I remember that announcement. I remember thinking, oh, wow, I didn't know Martian asteroids could get to the Earth, and let alone have their bacterial fossils survive. And that was a very big deal. What I didn't know at the time, at that age, is that it was actually very controversial. And right after, we learned that there were a lot of interesting things going on. Actually, when I went to that astrobiology conference in 2001 or so, I remember the debate there. And the debate was essentially saying that, look, just looking at stuff, this is not enough to say that that's a bacterial fossil. And one of the pieces of evidence used to say that those were fossils was the amount of carbon in there. Now, after that long, carbon isn't in the same form. You won't see the molecules exactly like they looked, like some of the molecules I was showing you. All sorts of processes will decay those things over time. They won't be recognizable in that way, but there's carbon there in long chains, and the way you typically see it is polycyclic aromatic hydrocarbons. So you get a lot of these hexagons that I was showing you earlier. But the trouble, so you'd say, oh, well, that's got to be life. That's how you get those on carbon chains. That's so characteristic of life. But it turns out those chains can form similar forms, not quite as complicated, but you can get carbon chains through what we call abiogenic processes. One is known as Fischer-Tropsch. So in very hot conditions, carbon dioxide can form these hydrocarbon chains, which don't have to come from life. Another piece of evidence they used were magnetite crystals. And this is supposed to be pretty good. There's apparently bacteria produce magnetite in sort of a string of pearls kind of way. And it was McKay's own brother that said, no, I don't, I don't think that's true. You could do this without bacteria. And even one of the other big things was that you could get this morphology without using biology. Some people were demonstrating that if you just sort of cook the rocks in this way, you would be able to see those kinds of patterns. And so there was a big controversy about this. And if you look today, I have some links at the end of the presentation that you can follow.
Mars.
And so don't worry, it's okay. That's fine. This morphology, it's not obvious that this is alive or this was a bacterial fossil. And if you look today, I think the consensus is that I don't think we can say that it is, that as much as we'd like to believe that this was evidence of a bacterial fossil, we don't think it wasn't. This happened right in 1996, just before 2 Mars missions were about to launch. And I remember this. I'll just briefly show Bill Clinton giving the announcement here. Tell me if you can hear it. I'll turn on my volume just so that. And we're just gonna. I don't want to make us listen too long a video. It'll just be for just 30 seconds or so of it. There he is. Okay. Someone wants to give me a thumbs up once he starts talking to hear it.
Good afternoon. I'm glad to be joined by my Science and Technology Advisor, Dr. Jack Gibbons, to make a few comments about today's announcement by NASA. This is the product of years of exploration and months of intensive study by some of the world's most distinguished scientists. Like all discoveries, this one will and should continue to be reviewed, examined, and scrutinized. It must be confirmed by other scientists. But clearly, the fact that something of this magnitude is being explored is another vindication of America's space program and our continuing support for it, even in these tough financial times. I am determined that the American space program will put its full intellectual power and technological prowess behind the search for further evidence of life on Mars.
So it's a longer announcement, but it just shows you the geopolitical importance of these sorts of discoveries and how they're used to sell what, to fund a space program. You have to keep your public very happy paying for it. And when this discovery was announced, no one wasted any time in telling the public how great a thing this is. And he was careful in his remarks too, right? He was pretty hedgy about it. He was saying, well, this has to be confirmed further. He was clearly been advised that this isn't a controversial finding, but they were very quick to jump on it and project that to the public. So I think Alan Hills was not a fiasco per se. It was a bold guess and a lesson in how difficult it is to determine whether you were able to observe. How do you look for life outside of Earth? It's not even obvious that you can do it even when the rock is sort of sitting right on your doorstep. And what sort of things you need to be able to do that. So that was our first one. And I think today the consensus would, one has to be careful, but the consensus today would be that we don't think that Alan Hills was good evidence for life on Mars. Does anyone recognize this picture? This is a little different. I don't think it's as canonical as Alan Hills. So this was, I don't know if anyone remembers it, in 2010, there was a NASA made an announcement. And I remember every step of it. I like the buzz, I can't, whatever you think, if you don't remember, whatever you think the buzz was around that time, it was bigger. It was the biggest buzz that, NASA never makes an announcement and keeps people on edge and has a press conference with tons and tons of people. This was just the, biggest things. I just can't emphasize what a big deal this was. So NASA had seeded some buzz, and when they made the announcement, a group of scientists got on TV with a really, really big press conference claiming that they had found a bacteria in a place called Mono Lake. That's in California. There's a lake there that's very rich in arsenic. And yeah, that's right, extremophiles, right? And that could survive in that lake. And the claim from the scientists was that this bacteria was replacing arsenic for phosphorus in all its major biomolecules. So we saw phosphorus back here in DNA. Where do I got it? Where was it? Here. That phosphorus atom, they claim that phosphorus atom was being replaced by arsenic in this organism. Now you may think that, well, okay, sure, what's the big deal? Like if you look at our amino acids, sometimes you can replace, for instance, oxygen with sulfur. You can even replace that with selenium, depending on how you do it, sometimes very little consequence. So arsenic, but that's not some great leap. But as a chemist, when you look at it, I've got it right here. So they thought that's where the phosphorus was. That's arsenic, whereas that's now that's arsenic now, where normally it's phosphorus. As a chemist, when this happened, I can't tell you what a big deal I thought this was, and so did everyone. I can show you. I didn't include everything here because to sort of emphasize what a big deal was. I can just tell you that whatever you think, it was bigger. Everyone was just expert chemists were writing this up, just going nuts. It was like the second coming kind of thing. So it was a big deal. And when you look at that, the first thing I asked everyone sort of nervously looked around and said, "Oh, that's arsenic. That's neat. I didn't know you could do it." And I said, "Did you get, there's a sort of evidence that you can get called a crystal structure, which is sort of like a 3D model of the molecule. So they must have gotten a crystal structure." So no, they didn't get a crystal structure. They sort of did it through these other more indirect means. I said, "Oh, okay." Like, why didn't they get a crystal structure? And then there's all these people chatting, being like, this doesn't make any sense. And then the chemists start saying that, wait a minute, this stuff is not stable. Like, we've never made this molecule in the lab. But based on what we know about arsenic chemistry in very, very similar contexts, this thing should fall apart in seconds, in really tiny amounts of time in water. It should not be at all stable. And then everyone started asking questions, and then there was a long period where it became very clear that this was a fiasco. This was just wrong. It wasn't even, wrong in the most naive way. And as things went on, this was within 24 hours of the announcement. It was such a weird thing because you had NASA certifying this. Like, this press conference was such a big deal. I'm just going to play a little bit of it here again, just a few, maybe about 30 seconds to a minute, just so you can remember what that was like.
Distinguished.
And keep an eye on this fellow, Stephen Benner. I met him at a conference. He's a chemist. He's a really smart guy. And I'll just play the clip for a second, and then I'll show you. So,
Foundation for Applied Molecular Evolution, Gainesville, Florida. Pamela Conrad, astrobiologist, NASA's Goddard Space Flight Center, Greenbelt, Maryland. And joining us via phone from Tempe, Arizona, James Elser.
like, just showing you this was a big deal. The world press was there. This is like the vision of what every scientific discovery is supposed to look like. People calling and say, I'm so-and-so from whatever newspaper in Spain. This was not a small deal. So I'll just show you the beginning where the scientist who was responsible for this started talking.
Dr. Felisa Wolf-Simon, the lead author on the paper, to fill us in on what she did and what she found.
Thanks, Mary. Well, as Mary probably knows, and as many of my colleagues would agree with, I'm always interested in exceptions to the rule. And what I'm going to talk about here today is not that much different than another exception to the rule. And so I've led a team that has discovered something that I've been thinking about for many years. And I've been thinking about an idea of substitutions. And what does it mean to be substitution? And what does it mean to be toxic? So I've led a team that has discovered a microbe that can substitute arsenic for phosphorus in its major biomolecules. But let me step back for a minute. All life that we know of requires carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And it uses those 6 elements in some of the critical pieces I think we're all familiar with, including DNA and RNA, or the information technology of the cell, the proteins, which are the molecular machines, and the lipids, which separates you from everything else.
So I'll spare you that. And I think there's a part, right? This press conference is like, I can't remember, I think it's like 3 hours or something. It's unbelievably long. And like I said, it's just I don't know what you want to call it. The enthusiasm is just like nothing you've ever seen. And so they have these panel members up here, and I think I got it at the right spot. And I want you to I've just got a clip of Steve Benner here, and I'll explain what he's doing in just a second. See if I can watch it.
Absolutely, to illustrate the point, if you're not so familiar with seeing those molecular structures, I think what we've presented, the parsimonious way to interpret this, arsenic was absolutely associated with the DNA fraction, purified on a gel. To address what Steve's saying, how it survived all the manipulation, if you're aware of how we do DNA extraction, unknown. It migrates at a different level. I'll be a bit technical for a second.
I think I couldn't get the right time point. I'm just gonna see if I can get Stephen. There's a beautiful piece. Well, I can.
Steve Benner in the room.
There, here we go.
I got to interrupt you, Steve. I'm sorry.
Rise for the
Here we go.
Tectonics. Generally been with someone as esteemed as Steve Benner in the room. I've been working on the next paper, should be submitted in February.
Maybe we can turn the question back to our colleague from Nature. I mean, the question is, you've taken a biochemistry course, and what do you remember from your metabolic pathways that you were taught, forced to memorize for the examination, and perhaps promptly forgot? So when you biosynthesize DNA, right, the phosphate comes from a nucleoside triphosphate, where the phosphorus that ends up in the DNA is the one directly bound to the deoxy-ATP, deoxy-GTP, and so on. And that phosphorus gets to that species by a 17-step metabolic pathway, which I'm sure you can write out on a piece of paper.
I got to interrupt you, Steve. I'm sorry.
So I'll spare you the rest, but what the dynamic through that entire thing, it's really worth watching. And Steve Benner, like I said, he's a well-known expert organic chemist. He could see through this from the beginning, and he'd clearly been carted up there, and he had no choice but to kind of smile and politely say, yes, I'm not sure if this is actually true. And deep in his heart, he knew what was going to happen, and he was probably trying to save all these other people from great embarrassment. And so he has a very tactful tone throughout the entire conference. And but just unbelievable kind of thing that that this could happen. That paper remains unretracted today. The scientists still stand beside it, behind it, and we know it's not true. Everybody knows it's not true. It's just a catastrophic failure of science. And science as a public is such an important thing, especially today. There's a lot of talk about vaccines and everything like that. It's important for the public to be able to trust scientific authorities, especially when they go out and give a press conference and do this kind of thing. And when you have these kind of catastrophes happen, I don't blame. I have a hard time being critical of those who are critical of the institution of science, because you can have these catastrophes happen. You may wonder, well, how did that paper get accepted? All this peer reviews, all the peer reviews were glowing, right? I mean, this is just it's every possible catastrophe put beside each other. They wrote a paper that made no sense. They got reviewers that did not understand the importance of how remarkable a discovery it would be, how extraordinary it would be, and they approved it, and that got published. And none of the people involved has ever sort of apologized, expressed any remorse for doing this. And so, I will never forget this. My friends always, I bug my friends, we talk about it all the time. But a lot of us, I think it's just left public memory, and it's just a catastrophe, and it's a fiasco. And part of what they were trying to push there is because it was part of NASA's astrobiology program, was that this was important, obviously, to understanding life on other planets and other things. If maybe an organism somewhere else could use arsenic instead of phosphorus, but it had no such thing. I could spend 5 hours just talking about this one incident. There's so many juicy details in there, and how it was disproved and everything like that. And it's just a remarkable story that's not appreciated as much. Just to show you some other fiascos, this happens all the time. There was an incident in 2001 where Kerala, India, there was a red rain, and what happened after that it's hard to explain. This guy here, Chandra Wickramansinghe, he's a Sri Lankan astrobiologist, very legitimate. He had worked with Fred Hoyle. He was responsible for really seminal discoveries about the composition of interstellar dust, the carbon content into it, the organic molecules that we can find in interstellar space, that I'm going to show you some of them. Just a very legitimate scientist, extremely important work. He should be proud. And he was the first one to jump on this and say, these are alien cells, kind of thing. And you can see the kinds of things they're pushing at. It's not true. This was I believe, so was this in algae, or was it red sand? I can't remember off the top of my head right now, but there's a very terrestrial explanation for this red rain. And it's certainly, it's not very responsible for, especially accomplished accredited scientists, to jump up and say, oh, this is certainly alien life, kind of thing. And this Chandra Wickramansinghe, you can find multiple stories like that about him. Every single time there's something in the news that he'll jump out and put a press release or something saying that this is extraterrestrial life, and of course there's always a very terrestrial explanation. Just recently, there was an announcement about a protein called hemolythin, and it was claimed that they found this protein. This protein on, sorry, did someone ask something? I might have missed it. Okay. They found this protein in an asteroid sample, like Allen Hills, this team from Harvard. I could look at this right away and see that this was wrong. They called it hemolythin because it had iron and lithium attached in this weird way that doesn't make a lot of chemical sense. You can't tell, this is the tip of it. And what they had done is essentially a very naive, poor chemical analysis on that sample, thinking that they're seeing a protein with iron and lithium in some chemically implausible way. But this was parroted in news releases and stuff like that, not nearly like, nothing big like arsenic life. But this was very obviously a wrong kind of thing right away. And yet, you'll see people mentioning it here and there. So it's another fiasco. But the reality is that truth is really better than fiction. What we do know that's real about the molecules that we find in space is really better than any of these sort of made up things. I'll just go through a few of them. Dan Toffick's lab studied that arsenic bacteria that they were talking about, it really did survive on very little phosphorus. It's actually turns out to be very difficult to grow something under phosphate-free conditions that the group was claiming to do. So the bacteria is just very good at scavenging the phosphorus that is around and using it in its biomolecules, even when there's very little. And they discovered why it's able to do that. This is the phosphate binding protein on the left. It looks like ribbons and stuff, but it's because you can't, there's too many atoms to show. That's sort of the main backbone. What that protein's job is essentially to take phosphate and bring it inside the cell. And what they found is that they got an unusually high resolution structure of this protein, sub-angstrom. Typically, it's just by luck. It's very rare that you get a sub-angstrom level crystal structure of a protein, but that allowed them to resolve the difference when phosphate binds to that protein and when arsenic binds. And if you look at the arsenate, this is the phosphate group here that's bound to it. And this is arsenate. And what you can see is that this angle of binding is very, very stressed. It's not a good angle. It's very sort of high energy. So this is how this protein is able to scavenge the phosphate from the environment over the arsenic, whereas lots of us can't do that. Arsenic is poisonous to us. If you swim around in Mon Lake and drink too much, that's not very good for you. But this protein that this bacteria has can resolve between arsenate and phosphate very well. And that's a discovery that only came about from studying that organism. There's a really well-known experiment called the Miller-Urey experiment. Urey was his advisor, where essentially they just took water, methane, ammonia, hydrogen, and bubbled some water into it with some electricity in some sort of primordial pond with a lightning, and that's what it's meant to simulate. When you do that, what they found was that you could retreat, you get this tarry goop out that's actually very difficult to analyze. But when they do that and subject it to analysis, they could find some of the amino acids, that some of these essential amino acids that we found that all life uses. That's amazing, right? That you can make that these parts that are very characteristic of life, some of them, not all of them, and there's a debate as to which ones you could and couldn't make through that experiment. You can make just by sort of, cooking stuff together, some of the basic parts together. They form spontaneously, and they also form adenine, which is one major part of the DNA base that we were talking about. So that's a hint that however life formed, some of the parts that are just sort of the key parts of life, you can get surprisingly far just by cooking, which is a profound idea, which suggests it may not be as difficult as we think, even though we haven't found the answer yet, no compelling answer yet. These are some organic molecules that we found in the interstellar medium, in outer space. So you can see that carbon chains are not a unique property of Earth. We can see them in meteorites, or we can detect them in space. You can see these hexagonal rings that are common. The one interesting one that we just discovered in 2016 is this propylene oxide. And you can see two things here. And propylene oxide, so if you want to read it, these bold dashes are, these bolded bonds, or bolded wedges rather, are sort of coming out of the page, and these dashed wedges are sort of receding into the page. So you can see how these two molecules are left hands and right hands of each other. You can't, if you rotate them around, you can't get them to line up, but they're the same number of atoms and arranged in the same way. So propylene oxide is chiral, and this is the first chiral organic molecule detected in outer space, and that was discovered in 2016, done through sort of radio, I think it was radio spectrometry. That's really crazy. That's the first time we've seen a chiral molecule outside of Earth, and tons of it, stellar quantities kind of thing. So, but what we don't see is one more than the other, as you wouldn't expect to see that, because there has to be some sort of underlying asymmetry that would give rise to more of one than the other, an enantiomeric excess, as we call it. But these parts of life, these key carbon-based parts of life, they're out there. And how they come together, the main puzzle is how we make these chains. We can make the parts, but the long chains, like these protein chains, those are things that we haven't figured out what abiogenic process will strongly, we don't have good candidates. We have some candidates, but we don't have a strong candidate. So which one could possibly make these chains together that are very long without existing life. And just one last one, Floyd Romesberg, who's at the Scripps Research Institute, who's been fired since, let go for some very non-ideal behavior with graduate students, is what his lab had done, and this is a huge deal. They were able to, biologists have been doing some very interesting experiments where they were able to, there were 20 amino acids I showed you earlier. We always call it the 20 amino acids of life. There's a few more that other organisms use, but we don't really, we call those very, very tiny exceptions to the rule of 20 amino acids. Biologists have been extending that genetic alphabet for a while by essentially in this codon table, there's multiple 3-letter codes for stopping the protein. So what biologists have done is essentially take over one of these codons and incorporate all the machinery to add more amino acids than these original 20, which is miraculous, which is just amazing. And they can insert many, many different types of amino acids. What Floyd had done is discover 2 new DNA bases that look nothing like the DNA bases that are common throughout life as we know it. Every organism uses these DNA bases, from, like I said, from archaea to full-grown human beings like us. These things, if you told me these things were from Mars, I'd believe you. That's how different they are. Yet we could discover and engineer that. And what he was able to do is incorporate these bases. It's one thing to make them in a test tube. It's another thing to actually get them into a bacteria and having the bacteria use them from beginning to end to make proteins and stuff like that. That's profound. And they look nothing like the natural bases. And in fact, whereas the natural bases have these polar bonds, they don't have that. They're greased and they sort of stick together in a sort of shape complementary way. That's super profound. There are alternate bases that we can just stick into bacteria, and with some extra machinery and a lot of graduate student blood get working. But the fact that there's a bacteria sitting in a lab that can use DNA bases that look nothing like bases as we know them, that's mind-boggling. Like I said, if you were to tell me that was what we found on Mars, I'd say, yeah, well, that makes sense because it shouldn't look anything like us, and it made it differently, and that's how it did that. Yet that's a human invention. And the last quick slide, the other parts of astrobiology that people are looking, of course, exoplanets, right? We all learned about exoplanets in our lifetime. There were no exoplanets. There was a time, before and after exoplanets. Once we determined that there really are other planets out there, we suspected they were there. How do you tell if life is there? It's very difficult. Some of the ways that NASA astrobiology is looking at are look for oxygen. All the oxygen on Earth is biogenic. It only happened when cyanobacteria-like things were producing oxygen through photosynthesis. Before those bacteria, there was no gaseous oxygen on Earth. Looking for chlorophyll. Do we see that chlorophyll has a certain color that's optimized in some cases for the spectrum that the sun emits? Can you see that? It's called a vegetation red edge. Can you see that on the reflectance spectrum of a planet that's very, very, very far away? Can you see some of the characteristic pigments that we see on Earth? Can we see chirality? Like I showed you these chiral molecules. These chiral molecules can have very specific signatures. And so if we can detect just the fact that does a chiral molecule exist on another planet, does that give us a hint that there might be life there? So it's a really tough problem to figure out, is there life sitting on another planet when you can't sort of go there and walk around? So, and I've just put in some links to some of the papers that that support all these things. All the images are linked to their underlying sources. And so if you want to learn more about those, you can click on those, but that's it. I know that was really fast, but I just thought better to rush through it a bit. But happy to talk more about it.
What do you say? Maneesh?
Yeah, go ahead, Mr. Clayton.
I'm wondering how much can light transfer all this information from a source beyond Earth to understand what we're looking at?
Yeah, that's this, I think these exoplanet biosignatures, what NASA's been looking for. It's under the context of, obviously you don't have a lot of resolution, right? Like the planet's very far away. So there's going to be all sorts of masking and things like that happen. So what sorts of assumptions can you make that might be able to tell you something specific? And the thought, there's no answer to that. There's no clear answer to that. Maybe it's impossible to detect if there's life on the planet. But these are some of the ideas that people are looking at, that there may be a good enough signal to noise ratio that we would be able to pull them out. If you really saw this vegetation red edge, the thought is that if you could detect that, there's a good chance that it would be able to stand out against all the noise factors that you're invariably going to run against.
And we're hoping that the James Webb telescope is going to answer a lot of those.
Yeah, exactly. Some of those
questions.
things. Yep. Yeah, exactly.
Right.
And so that's and then the question is, are we going to see a biosignature, or are we going to get a call on our radio telescopes first? Nobody knows, right? It's like, but these are just this is really such a hard problem. You can just imagine how much expertise you have to run a credible project to even try this, right? You need people that understand planets, people that understand atmosphere, people that understand biology, chemistry, like you just need a big group of really, really smart people. And one thing I meant to finish up on was that when you see all those fiascos, how do you explain that, right? What's going on with everyone that's doing? It's a very simple explanation. It's that this is how the Institute of Science runs. These large agencies, they choose what to decide to fund, and scientists, they're just like guppies waiting to grab the money to go do the thing that the agencies want to do. And you can see when you make people too desperate, you get stuff like this. And it's a bad thing to do, I think, right? It's everybody, some of those fiascos that I showed you, that I think they're displays of, like I said, very terrestrial behavior, very easily explained. It's scientists that, Felisa Wolf-Simon, she gave that bacteria the name GFAJ1, which stands for give Felisa a job. And I mean, it's funny, but it really does reflect the position that many scientists find themselves in, and there's a desperation to it, and you can see why these people there's no benefit in admitting that you're wrong. There's no benefit to fixing things, and so that's probably how that works.
Yeah, I always one of my favorite sort of sayings that has come out of the history of science, it comes from Richard Feynman, and he said the first principle is you must not fool yourself, and you are the easiest person to fool.
Definitely.
Meaning you are the easiest person for you to fool.
Yep.
We want things to be true, and we just are not capable of sort of guarding against all of the little biases and things that can influence our decision making and so on.
Definitely, especially when there's such a big prize. This is the origin of life, or life. This is the most profound idea out there, right? And so the calculus is that, well, if I get to do something, and if I'm wrong, well, there's no real cost to it, right? You're in the same position. So that may not be a good way to run your science infrastructure kind of thing. So
you mentioned exoplanets, and we know of thousands of exoplanets around stars in the Milky Way, but we don't know of a single extragalactic exoplanet. And the reason is simple. Basically, they're too damn far away, right? We can hardly even see the difference between two stars, let alone anything that's actually. But methods might be developed in time that will allow us to do that. But the point is, as far as we know, the Copernican principle, or the principle of mediocrity, still applies. Everything we observe here in the Milky Way, we have no reason to believe that it's special or unusual. There's no reason to believe that it isn't exactly, the statistical occurrence of certain things isn't similar in every other galaxy that's out there.
Right.
I always like to look at something that Neil deGrasse Tyson said, where he said, if you look at our analysis of the distribution of elements in the observable galaxy, I might get the order wrong, but there's hydrogen, oxygen, carbon, nitrogen, and I think one of the inert gas
Helium.
comes next. Helium, of course, yes. The inert gas, helium. Anyway, yeah, they're the most common elements in the whole universe, and they're what we're made of, except for helium, of course, but it's inert. That's why.
Right.
So you take that sort of fact, and then you look at our detectable history of life on Earth. The very earliest detectable signs of bacterial life on Earth occurred within the first couple million years of, as far as we can tell, the origin of Earth 4.5 billion years ago or whatever. So it's almost like it's inevitable. I mean, there's no reason to think that the same kind of thing hasn't happened multiple times given the right circumstances, the right materials, and a stable enough environment and an energy source and the blah, 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 blah. So yeah, we can't, we haven't found it, we haven't proved it yet, but
Yeah.
there is a lot of circumstantial evidence that it's out there. So we shouldn't stop looking.
Absolutely. Yeah.
I'd like to add that I don't know how many of you have seen in this month's or the new November, December Sky News. There's an article in here by Ivan Semenek about Canadian astronomers actually trying to figure out what the light that they're getting from these exoplanets could hold and what they can see in it. So have a look and read the article. I haven't finished it all yet. It's a pretty good article so far.
Yeah. The first confirmed exoplanet around a main sequence star was discovered in 1995. That's just yesterday.
Yeah.
Relatively speaking. And we now know of about 4,800 confirmed exoplanets in hundreds of star systems. Sorry, thousands of star systems, hundreds of which have multiple stars. It's estimated that 1 in 5 confirmed exoplanets is a Earth-sized sort of planet within its star's habitable zone. So, the conditions are proving to be sort of right, but we just don't have the technology yet to be able to sort of detect the signs that confirm that there's life out there.
The other thing about that too, though, is like there could be a lot more planets than that, because it all depends on whether those planets actually pass between the star and in the direction that we see from. If it doesn't, let's say it's orbiting in an orbit around where we can't see it, like the star is here and the planet is going here, it never crosses across in front of the stars. We don't see the planet. So there can still be a lot more exoplanets than even what we've found so far.
Yeah, no, well, you're right that there undoubtedly are more that we haven't detected yet. However, when it comes to methods for detecting, there are dozens now, and a lot of them involve, just detecting any kind of spectral, Doppler shift indicating a wobble in a star's motion. The planetary companion does not have to be, in a line of sight or, passing between us anymore. They've developed so many different ways of detecting exoplanets, but yeah, the science for actually examining them now is what needs to be sharpened up.
Well, a good example is us as amateur astronomers try and get a good picture of the planets. These people are looking at planets that we would never even imagine seeing and figuring out what they're made of. So their instruments are far more sensitive than ours.
Yeah, so it's a fascinating subject. I think it's the question that it's like the ultimate question, isn't it? The answer is not 42 either. It's whatever, but
where's James T. Kirk when you need him? I'm just saying, send him out there and, maybe we'll get something back, engage number one, I'm just saying.
An amazing.
Anyway, this has been a very interesting topic for sure. Lots to think about.
Yeah, there's a lot in there. I sort of like the fire hose approach a bit better. I hope there's enough, fairly modern new ideas in there that there's a good chance I think you haven't run into before with two-domain structure of life, chiral molecules that are out there, are unnatural bases and amino acids that we were engineering into bacteria today. All that taken together is pretty neat new stuff kind of thing. So,
well, I think just based on the conversation, it's opened up a little bit of a rift here for people to talk about. So,
yeah, it's interesting. That's for sure.
Yeah.
Also makes you wonder: is carbon the only way of making life? And we still don't know. We still don't really have that answer.
Right. If you begin, like, what happens at very different temperatures, things could be totally different. You could, all these things that are unstable at room temperature under standard conditions might fly out the window. Maybe you can make long arsenic chains or whatever at minus, 200 degrees Celsius or something. But
I mean, even gravity, like a higher gravity on a planet.
Sure, yeah, or super, super high grade. But the best we know is we have, like, it's not a claim that we know for sure it's this. It's just that you have to be talking about carbon chemistry, because once you start talking about, if we're talking about life outside of carbon chemistry, then that's something very different, something that almost impossible to imagine. Doesn't mean that it can't exist. It just means that anything we think about, if we're going to build experiments to look for things, we sort of have to say, well, you got to put some boundaries around it somewhere. And we say, look, the carbon chemistry as we know it is essentially, what defines life. And if there's something else out there, well, who knows then, right? Like, I mean, we don't even know how to look for that then.
Yeah. So basically designs experiments to find what we can prove and what we do know.
Right.
So that when we're looking at things that are that far away, that we can prove without a shadow of a doubt that yes, that is life.
Right. Exactly.
Do you think really we can prove anything beyond a shadow of a doubt? Because quite honestly, a lot of it is guesswork and it's just what we think is right.
The weight of evidence, in other words.
Yeah,
theory and speculation.
Yeah, a lot of it is guesswork, theory, speculation. And I don't think we're ever going, well, not in our lifetimes probably, because a lot of us are bloody old.
Unless ET phones home and talks back to us.
Yeah.
How about aliens? What are they made out of? Phosphorus, silica.
But I mean, our basic chemistry says that it's not I can say for sure. It's just that if there is a life form that is made out of something not carbon based,
right,
then it sure is different than it's just it's not even something someone can propose, because as I said, these sorts of molecules are as we know it, are no other element formed stuff like this, at least under conditions as we largely know them.
Right.
So to have life, you have to have these sorts of shapes, and to have these sorts of shapes, you have to have carbon under these conditions.
And
so
we sort of understand the reasons for that too, don't we? Because it's got to do with the number of valence electrons and things that make themselves available for bonding with other molecules or atoms and so on.
The strengths of those bonds, they have to be certain. So carbon-carbon bonds have to have a certain strength. So, like, for instance, there's other, you might have remembered, if you look down the column, you've got similar sorts of the atoms have similar sorts of properties. Like fluorine, chlorine, bromine, they're all halogens. They have similar properties. They're all noble gases. Carbon, you've got silicon and lead there too, right? Those things don't, despite having similar properties in one sense, they're even outside of life. We can't coax them into shapes that sort of look like this. This sort of structures of discrete molecules that have specific properties. That doesn't mean it's impossible. It just means that you're just talking about something that so far, if one is going to say, well, then what about silicon-based life? Well, then we should be
Well,
able
yeah,
to silicon.
that's one of the messages that got sent through. What about silicon-based life?
Oh,
Like,
right. Sorry, I missed that. Sorry, I wasn't looking at the chat.
you know, so yeah,
Right.
carbon-based life as we know it, well, as we know it now, is the base for our life, right?
Right.
But
And
what about silicon-based?
yep. So silicon is right under carbon, so it's similar. Now, the
It's
explanation.
also, I should mention, Maneesh, silicon is the next element in that hierarchy of quantities in the universe.
Right.
Yes, there's a lot. As you know,
It comes right after whatever I said before.
definitely.
It's the
I mean,
fifth one, yeah.
yeah, one just
Right.
needs to look outside. A lot of our Earth seems to be sand, and that's, of course, the most abundant element is oxygen, and then because there's two oxygens for every silicon, so you have
Silicon, yeah.
Sorry.
I say, yeah, silicon-based anyway.
So right. So when we say silicon, it's not that it is impossible. No one can say it's impossible. It's just that when you think about the way silicon chemistry on its own outside of life, even like as we saw, even without life, we can make things like urea that come from life, right? Silicon doesn't have, to our knowledge, anything that resembles this kind of diversity and shapes. You can make long polymers out of silicon, but
Right.
can't quite make anything that has this as far as there's no analog as far as we know. So it could exist, but before we put a strong bet on silicon-based life, we'd almost want to say, well, can you make a diversity of molecules outside of whatever the silicon life? And then if you could, then you might have a good hypothesis. But right now, we don't know that that's true. And it does relate back to silicon bonds being weaker and all sorts of things like that. But without thinking too much about the basic properties of silicon, you can know empirically that there are no silicon analogs to these types of molecules that exist. You can make molecules out of silicon and neat things that nothing like this.
Basically, you're thinking outside the box.
Right. If they exist, then that's going to require some really new principles on just basic chemistry rather than
right.
It's an easy question from the perspective of chemistry. There's silicon molecules don't look like carbon-based molecules as we know.
It's principles that we don't really understand right now.
Right.
Yeah, or maybe another way of saying that is that it relies on maybe conditions that we have no evidence exist.
Right.
Going back to that Copernican mediocrity principle, why would we invent conditions to support a theory that we have no evidence that they exist, and we have a great deal of evidence that are not found in almost everything we've ever seen? So, yeah, it seems tremendously unlikely, and I think it really dates back to sort of some science fiction author in the 50s or something, or the 40s, who probably first proposed the idea based on his limited knowledge of.
Right. I mean, it's a very, very reasonable idea, and it's not always explained really well that carbon seems special because of its chemistry that we can do just with our own hands and salt of the earth. Where silicon does have its own chemistry and its own compounds, it's not like we can say, well, we can make the silicon ones and somehow life chose carbon. Doesn't seem to be the case. It seems that carbon's chemistry is special and unique and suited for this kind of thing.
Okay, so just a timeout here, just to do a 360. I'd like to thank you for your presentation. It's definitely been an intriguing presentation. It certainly got all of us thinking.
I'm so glad.
And I'm going to stop the recording now. I'll leave the session open for anybody that wants to talk after that. I'll still say stay here and whatever, but I'm going to call the meeting itself officially at an end. And yeah, anybody who wants to talk, then sure. It's all open, and we certainly appreciate it, your time, and you gave us a lot to think about.
I'm so glad. Yeah.
Yeah.
Okay.
Yeah.
Same as last time, it was like afterwards, last time, we all emailed back and forth to each other and said, okay, well, like what? Yeah, what do you think? You know, so
Good. I'm so glad.
I still think you're adopted because, you know, but no, we
Oh, God.
definitely appreciate your input here, and I'm pretty sure, I can pretty much assure you that we're gonna have you back again.
Oh, we'd love to.
Always
Yeah.
love to. Yeah.
So yeah, it was